Hi, good day, everyone. In our previous revision class, we talked about how to determine the amino acid composition within protein. Prior to that, we've shown how to isolate and verify a protein. So we'll build up from there. After you have isolated and verified your protein, remember I would say you can actually collect or excite or remove or put out the verified protein and put it in a buffer or a solution. Inside the buffer or solution, in a test tube, you can carry out sequencing to determine the order of those amino acids and protein. How can you know? This is what we will be discussing today. Now, to start our discussion, peptide can be sequenced using basically two ways. There is either Sanger sequencing method or using Edman degradation. In Sanger sequencing method, Sanger uses the reagent called 1-fluoro-2,4-dinitropentene. This is the first proposed method to determine the amino acid sequence. Even though it wasn't successful, but a tremendous achievement was realized. What was that? The Sanger's reagent, which is 1-fluoro-2,4-dinitropentene, was able to react with the terminal amino acids. When it reacts with the terminal amino acid, the next reagent helps in hydrolyzing the peptide bonds. So unfortunately, it hydrolyzed all the peptide bonds. But that first terminal amino acid that is being attached to 1-fluoro-2,4-dinitropentene can actually be identified. But if you identify this, you cannot identify any other amino acid again on that sequence simply because you have ended up in hydrolyzing all the other peptide bonds. Therefore, Sanger sequencing can only be used to determine the first amino acid or the N-terminal amino acid. In a situation where your protein is an oligomer containing two polypeptides, you are going to have an idea that this protein contains two polypeptides. Why? Because you will detect two amino acids serving as the N-terminal amino acid. If the protein is a trimer containing three polypeptides, you're going to detect three N-terminal amino acids. If it is tetramer, you will detect four. If it is five, you will detect, as a pentamer, you will detect five N-terminal amino acids like this. So at least you can use Sanger sequencing. If you want to know whether the protein is actually an oligomer. Other reagents like dancyl chloride or dapsyl chloride can also be used in place or instead of 1-fluoro-2,4-dinitropentene. So this method, in summary, can only be used to identify terminal amino acids and the number of peptide sequence approaches. So you can tell which amino acid is the terminal terminal amino acid, and you can tell how many peptide sequences are present in this protein. That is the Sanger sequencing. Now, we move to the next method called Edman degradation. In Edman degradation, Edmonds uses a reagent called phenyl isothiocyanate, also called Edman reagent. This phenyl isothiocyanate reacts with the terminal amino acid residue. When this happens, the next peptide bond of this terminal amino acid can be selectively cleaved. So this reaction occurs in an alkaline condition. So when you have phenyl isothiocyanate reacting with the N-terminal amino acid, you produce a PTC adduct, which is phenyl thiocarbamyl adduct. This phenyl thiocarbamyl adduct, in the presence of trichloroacetic acid, That particular peptide that is holding peptide bond that is holding the N-terminal amino acid alone can be clipped or can be broken. Once it is broken, the peptide chain can be released, while this adduct will now be released also alone. So the adduct can be converted. Initially, it forms anylothiazolinone. This anylothiazolinone can, in the presence of acidic environment, converted to phenylthiohydantoin. So phenylthiohydantoin can be converted or derivatized to something that can easily be isolated and then detected to determine which amino acid is that. When you determine which amino acid is that, now you go back and react with phenyl isocyanate again, bind to the N-terminal amino acid in the presence of an alkaline condition to form this phenylthiocarbamyl adduct. This phenylthiocarbamyl adduct in the presence of trifluoroacetic acid causes the release of the peptide. Now you have the peptide with N-2 amino acids. And then you now have the derivatives, and the derivatives can be derivatized to be isolated and determined or detected. And then you also react cycle until you're able to identify all the amino acids in that peptide. But the question is, sorry, this is the other one. But the question is, what is the limitation of standard Edman degradation? Edman degradation, you can only be able to determine the sequence for a short fragment of a peptide. You cannot determine a long polypeptide sequence. Now, if you want to determine a sequence, the amino acid sequence for a long polypeptide, how are you going to do that? You're going to do that through 4 different steps. Number 1 step is breaking of disulfide bond. So remember, if you are dealing with a very long polypeptide sequence of amino acids, or rather, a long amino acid sequence, also called a polypeptide, there is all the chances that this amino acid will definitely have to be folded into a tertiary structure. Now, once a tertiary structure is formed, one of the most important driving bonds that hold a tertiary structure together is the disulfide bond. And disulfide bond is very strong, and it will interrupt your process of determination of the amino acid sequence. So to make things easy, you just have to guess that in case if there is any disulfide bridge, let me break this disulfide bridge. How do you break the disulfide bridge? Breaking the disulfide bridge, you can use a 2 approach. You can either oxidize the disulfide bridge using palpomic acid to form what we call cistic acid. So this cistic residue are stable. They will not go back to form a disulfide bridge again. Or you use a reduction approach using dithiothiotol. When you use dithiothiotol will return this cysteine residue or cysteine in disulfide bond to cysteine. But this cysteine, remember, have the potential of coming back again to form a disulfide bond. So you have to prevent that. How do you prevent that? You add iodoacetate. This iodoacetate will form a kind of an acetylated cysteine residue. So you have a cysteine residue that will attach to acetic acid. So you have methylated methylated carboxymethylated cysteine. So this carboxymethylated cysteine cannot come back again to form a cysteine or to form a disulfide bond as in cysteine. Therefore, you succeeded in breaking this disulfide bridge. Now, after the breaking the disulfide bridge, the next thing is you can create a fragment. You can create several fragments. How do you create several fragments? We know that Edman degradation cannot sequence many. So if it cannot sequence a large peptide sequence, Then you break it into a smaller fragment. How do you break it into a smaller peptide fragments? Using a number of chemicals or enzymes. We can see trypsin can be used to break a long peptide, where it's a break a long peptide anywhere where there is lysine. So the carboxyl group of lysine, it can be cut there, or carboxyl group of arginine. We have submazlarose protease, which is another enzyme that coats the carboxyl end of arginine. We have chymotrypsin that coats at the carboxyl end of phenylalanine, tryptophan, and tyrosine. We have Staphylococcus aureus B8 protease that coats the carboxyl end of aspartic acid or carboxyl end of glutamic acid. We also have many of them like pepsin, endoproteinase. We have cyanogen bromide that cuts at the carboxyl end of methionine. So wherever methionine is found, the carboxyl end of methionine can be cut by cyanogen bromide. So you can use that to create the fragment, and then each of these fragments can be used to Edman degradation can be used to sequence them as we explained using phenyl isothiocyanate. Then the next thing is to order the overlapping sequence. So when you are doing the second step, which is creating the fragment using this reagent, when you use one particular reagent on a particular polypeptide, you can get several sequences. Like you can retrieve 10 sequence fragment. So you can decide to say, okay, I'm not going to use only one enzyme or only one reagent. You can use 2 or 3 or even 4 sometimes. So each of these will have its own fragment. So you can sequence the fragment. After sequencing the fragment, now all the fragment after sequence, you know them, they are sequenced. Now the next thing you will do is you consider, because they are all from the same protein. From the same protein, now you look at them, you compare the sequence produced, and then you'll be able to see where an overlap occurs. From there, you will be able to sort out the exact sequence of your amino acids in your sample. As we explained in the class, I know this is not a difficult thing. This is something you can easily be able to do it in as far as the polypeptide chain is not a very huge amino acid sequence. And like I said, question can come where the sequence can be given, the name of the cleaving agent can be given, cleave it and show us the fragment you will be obtained, or the fragment will be given to you from 2 different reagents or 3 different reagents, and you'll be asked to construct the sequence of the amino acid based on those fragments. Then you would use the knowledge of the overlapping sequence or the matching sequence to construct. That we already took it out the importance. Like I said, you have any question, please pause your question through the class group. It will get to me and you'll get your answer. Thank you.